Welcome students back to chapter 23 on the digestive system. Now that you've had a little overview of the GI tract, we're going to talk a little bit more about the functional anatomy of the digestive system, so a little bit more in detail about what goes on in each of the organs of the GI tract, as well as some of those accessory organs. So we are going to start, of course, where the GI tract starts, in the mouth and the oral cavity. So the oral cavity is pretty much banded by the hard palate above, and then the sort of tongue and other sort of soft tissue below, and of course the lower jaw. Your tongue is found here in the oral cavity. You've also got all of your salivary glands and your teeth. In terms of what goes on here in the oral cavity, this is, of course, our ingestion point. This is where food actually gets put into the system. And then there's a little bit of mechanical digestion. Particularly what we are talking about is mastication, chewing by your teeth. That's why you've got all those nice, lovely teeth. In terms of chemical digestion, anybody big fan of maybe, say, potatoes? Some nice yummy mashed potatoes, perhaps? They will start to be broken down here in the oral cavity. You have a little bit of amylase enzyme in your saliva that starts breaking down starches. And then there's a tiny bit of fat digestion that occurs in the oral cavity as well. In your saliva, there's also a little bit of lingual lipase, which is an enzyme that breaks down fats, but pretty minimal. Mostly what goes on here in the oral cavity is mastication by your teeth and that starch breakdown. In terms of our propulsive activity, what we are talking about is swallowing. Swallowing is a voluntary activity initially, and then once the food gets a little bit lower in the pharynx and esophagus, then it becomes a little bit more involuntary. The tongue itself is made up of skeletal muscle, so if you've ever eaten like cow tongue before, you're essentially still eating skeletal muscle, just as if you were eating sort of the other cuts of steak. The tongue has a couple different functions. The tongue plays a couple different roles in terms of helping you chew your food. It actually helps sort of mix your food and sort of mush it together, right? You know, when you're chewing your food, you sort of kind of push up against it with your tongue. The tongue is also really important in terms of speech. In order for me to even be able to talk to you guys right now, it requires sort of shaping my tongue properly. And of course, our tongue is where all of our taste buds are, on all of these little papillae that we find, little bumps all over the tongue. That's where your sense of taste is. So as you enjoy your steak and potato, or think of your favorite food, your ability to enjoy that is there on the tongue. And then the tongue also plays a role in swallowing. When you choose to swallow your food, you push your tongue up across the top of your mouth, and that sort of pushes what we call the bolus of food, which is sort of that mass of food, towards your pharynx. Our teeth are also found here in the oral cavity. They are, of course, is what is doing the mastication, the chewing. Different teeth as an adult than as a small kid. Our baby teeth, our deciduous teeth, come in typically starting about 6 months till about 2 years or so. And then we start to lose them when we're a little bit older, 6, 7, 8 years old. You have 20 deciduous teeth, but 32 permanent teeth. They do have different shapes. So the front couple teeth are the incisors. They are for sort of grinding your food. If you've ever seen a video of a rabbit eating a carrot, that's essentially what your incisors are doing, of little scissors at your food. To either side of the incisors are the canines. Those are the eye teeth, the sharp teeth. They're for kind of tearing and grinding at meat primarily. And then you've got molars and premolars. The molars and premolars are for grinding teeth. So if you've ever used a mortar and pestle where you sort of grind your food, that's what those molars and premolars are for. They've got these big tops, like round, we call cusps on the top of them, and they work great at sort of grinding your food. There are several small salivary glands, but three primary sort of big ones. The parotid gland, which sort of sits just in front of your ear on the side of your mouth, the sublingual gland under the tongue, and then the submandibular gland towards the bottom of the mandible. This, of course, is what is producing the saliva. Saliva is actually very multifunctional, helps cleanse your mouth. It helps dissolve your food so that you can actually taste it. In order for us to taste things, it has to be dissolved in something. It also helps to moisten your food, sort of compact it all together. And again, as I mentioned earlier, it does begin that sort of starch breakdown because it does contain that amylase protein. Saliva itself is mostly water. It is slightly acidic, so a little more acidic than our blood is. It contains generally lots of electrolytes, so lots of different ions. It will contain those two enzymes. You also find metabolic wastes in your saliva, some things like urea, uric acid, and you also find immune secretions. You have defensins, proteins, and antibodies in your saliva. So if you've ever, gotten a cut and taken a little saliva from your mouth and sort of washed your cut, it's actually a very useful thing to do because the saliva does contain some kind of antibacterial properties. Saliva and salivation is controlled by the autonomic nervous system. So just sometimes even the sight or smell of food will sort of activate the parasympathetic nervous system through its connection through the hypothalamus and the limbic system. So that you sort of start producing saliva even before you've eaten. Of course, there are chemo and mechanoreceptors in your mouth as you place the food in your mouth that also will activate the parasympathetic nervous system so that you start producing saliva. Conversely, the sympathetic nervous system inhibits the production of saliva, which is why if you've ever been really nervous, you've had sort of a dry mouth, because your sympathetic nervous system is sort of stopping that production of saliva. So you can see these little sort of what we call demi-moons. These are the cells that will produce the proteins, the enzymes that help break your food down, and then these mucus cells produce more of the sort of mucousy part of your saliva. The pharynx and esophagus, in terms of their functional anatomy, we really are just talking about a food chute. There really isn't much that goes on here in the pharynx and esophagus except for swallowing. So the esophagus itself, so let's talk about the pharynx first. Of course, we're skipping the nasopharynx part of the pharynx. We're just talking about oropharynx and laryngopharynx. When you swallow, the epiglottis will close over your trachea. That will route the food back into the esophagus, not down the trachea. And the esophagus really is just a big old muscular tube. It tends to be quite collapsed when empty and then sort of expands as food moves through it. The upper portion of the esophagus contains skeletal muscle because, again, initially swallowing is a voluntary action. And then the lower levels of the esophagus have a whole bunch of smooth muscle in them so that when the food gets lower, it's more of an involuntary function. At the end of the esophagus, there's a sphincter muscle. That's a little round ring of muscle called the gastroesophageal sphincter. And that generally will stay closed unless you are swallowing, and then it will open up to allow the bolus of food into the stomach. If you know anyone or have yourself suffered from heartburn, it's from sort of excess acid production in the stomach such that it sort of escapes that gastroesophageal sphincter and ends up in your esophagus, and that's where you're sort of feeling that burn feeling from. The proper term for swallowing is deglutition. It's actually an action that involves 22 different muscle groups, which is crazy just to swallow our food. There are two phases to swallowing. The buccal phase, which is sort of that voluntary phase, and then the pharyngeal and esophageal phase, which is more that involuntary phase, controlled in the brainstem using the primary parasympathetic vagus nerve. So this is sort of what that looks like. So you can see here in the buccal phase, the upper sphincter muscle is contracted, right, kind of closing off the esophagus. You've got that bolus of food, that mass of food in your mouth. You say, okay, it's time to swallow my food, and the tongue pushes up against the roof of the mouth. The uvula closes down, kind of blocking off the nasopharynx so that the food doesn't go into the nasopharynx and the nasal cavity. That then forces the food into the lower parts of the pharynx and ultimately into the esophagus. The larynx also rises. That closes the epiglottis over the trachea, so that the food is forced then into the esophagus. There are constrictor muscles around the pharynx, sort of just forcing the bolus of food down further and further. And then peristalsis pretty much takes it the rest of the way into the lower esophagus. The gastroesophageal sphincter will open up, and this food will end in the stomach. In terms of the stomach itself, it's about a 50 milliliter organ, but can actually expand up to about 4 liters of food. Think, full Thanksgiving, thanks to what we call rugae. So just like we had rugae and sort of folds in the mucosa of the bladder, helping it to expand, we see the same thing here in the stomach. Lots and lots of little folds in the mucosa of the stomach, so that the stomach can expand. There are a couple different sections to the stomach. The initial section of the stomach that connects to the esophagus is known as the cardiac portion of the stomach. The section of the stomach that sort of sticks up a little bit is known as the fundus of the stomach. Most of the rest of the stomach is the body of the stomach, ultimately leading into the pylorus and the pyloric canal, which ends at that pyloric sphincter muscle between the stomach and the small intestine. There are 2 curvatures to the stomach. The lesser curvature is the more medial curvature, and the greater curvature is the lateral curvature. The stomach has an extra layer of muscle in its muscularis. So in addition to the longitudinal layer and the circular layer that are sort of standard for all of our GI organs, the stomach has an additional oblique layer, so it is a quite muscular organ. The inside of your stomach is sort of a, shall we say, Mars-like landscape. It's not a sort of a happy place. It's pretty harsh inside the stomach. And so the stomach has what we call the mucosal barrier, basically this thick layer of very bicarbonate-rich mucus that helps sort of protect the inside of your stomach. So think like a thick coating on the inside of your stomach. There's also really, really tight junctions between all the epithelial cells of the stomach, so that stomach acid that's inside the stomach sort of doesn't escape and affect the deeper tissues. And stomach cells have a very, very high rate of mitosis because they're pretty much kind of constantly being damaged by the stomach acid. And so they repair themselves and sort of regenerate quite readily. Now, most bacteria do not do well inside the stomach. However, there is one particular type of bacteria that sort of does okay in the stomach. It's called Helicobacter pylori. So these little green bacteria that you see on the screen. And they are typically the cause of a gastric or peptic ulcer, which is sort of an erosion of the stomach wall where some of that stomach acid sort of, first the bacteria kind of breach the mucosal barrier, and then the cells start to get damaged, and then it sort of snowballs from there, and you can end up with part of your stomach wall eroding. In terms of what our stomach does for us, here's the fun fact about the stomach. You can actually live without your stomach. with the exception of one thing and one thing only. And we'll talk about what that is in just a second. Of course, our stomach is our temporary storage. So if you ever have known anybody who's had bariatric surgery or stomach staples, they have to eat in much smaller portions because they don't have that sort of 4-liter capacity. There's a lot of mechanical breakdown that takes place in the stomach. Again, there's that extra layer of muscle that sort of pummels and mixes the bolus of food into a more paste-like substance that we call chyme. There's lots of kind of mixing that goes on here in the stomach. And then there is the stomach acid itself. So it is hydrochloric acid. It is made by what we call these parietal cells right here. And the parietal cells make that stomach acid and also something called intrinsic factor. Hydrochloric acid is very, very necessary because our protein digestion starts here in the stomach. So the chief cells in the gastric pits create something called and secrete something called pepsinogen. And pepsinogen is an inactive enzyme. When the pepsinogen mixes with the hydrochloric acid, it becomes pepsin, and pepsin starts to digest our proteins. The reason we need protein digesting enzymes to always be secreted in an inactive format is basically if you had these chief cells full of pepsin, well, those chief cells are also full of proteins. They would basically digest themselves. So protein digesting enzymes are always secreted in an inactive format. That's what OGen always indicates, inactive enzyme. And then they require, and here in the stomach, the addition of the hydrochloric acid to turn them into pepsin. In addition to activating the pepsin enzyme, the hydrochloric acid also denatures our proteins, so sort of unfolds our proteins so that the pepsin can begin their chemical breakdown. There is a little bit of absorption that takes place in the stomach. We're primarily talking about things that are lipid soluble, like alcohol, as well as aspirin. And then the only sort of propulsive force that goes on in the stomach is some peristalsis and some segmentation, some mixing in order to deliver the sort of soupy chyme to the small intestine. And that does tend to happen in little spurts. So it's not like everything in the stomach, poof, goes at once to the small intestine. But I mentioned when we got to the stomach that you could technically live without your stomach, right? There are other parts of the digestive tract that break down proteins. There are other parts of the digestive tract that do absorption. But the one thing that the stomach does that nothing else in the body does is produce intrinsic factor. Intrinsic factor is this little sort of chemical that is completely and utterly necessary for your small intestine to absorb vitamin B12. And B12 is necessary for us to make red blood cells. And so theoretically, if you could get a B12 injection, you could theoretically live without your stomach, which is sort of a weird sort of fun fact, I think. Typically, you produce about 3 liters of gastric juice per day. It is controlled via the autonomic nervous system and then some hormones. So sympathetic stimulation is going to decrease and inhibit the production of gastric juices. Parasympathetic stimulation is going to increase the secretion of our gastric juices. The hormone that is stimulatory to the production of gastric juices is called gastrin. And then the inhibitory hormones are called enterogastrones. There are 2 of them, secretin and something called CCK, which are made by the small intestine. This is sort of what that gastric juice regulation looks like. So we've got 3 phases: the cephalic phase, the gastric phase, and the intestinal phase. We're basically talking about where we are, where is the food at this point. So in the cephalic phase, we're really talking about, okay, we haven't really eaten yet, but we're thinking about it, we're smelling it, we're looking at the food. So that sort of starts the process. the smell of the food, the taste of the food as it goes into our mouth. All of that, of course, activates our cerebral cortex. Our cerebral cortex can talk to the hypothalamus and the brainstem, which then activates the parasympathetic vagus nerve and releases more and increases the activity of the stomach. So again, sort of even before you've actually eaten anything, your stomach is sort of, and as you're eating things, your stomach is sort of getting prepared. It's getting ready to receive the food. As we move into the gastric phase, this is when the food is moving into the stomach itself. So there are stretch receptors in the stomach that sense when the stomach gets sort of stretched out. There are also chemoreceptors in the walls of the stomach that sense that there are foodstuffs in the stomach, that sense that the pH is and we're getting that sort of rising pH, so that we then say, okay, hey, pH is getting too high, we're going to start producing some stomach acid. That turns on sort of the hormones that lead to the release of stomach juices, and then the stomach distension activates both the long reflexes via the brainstem and the vagus nerve, as well as the short reflexes that result in the more production of stomach acid. The intestinal phase is as the foodstuffs, the chyme moves out of the stomach and into the small intestine. So you sort of have these partially digested foods in that first part of the small intestine, and there is a very sort of brief release of gastrin from the intestines that sort of just gives us one last little burst of stomach activity. In terms of inhibitory events, again, we can look at 3 phases: cephalic, gastric, and intestinal. Of course, if you are depressed, if you're sort of just not feeling well, that sort of loss of appetite can affect your cerebral cortex, which will decrease parasympathetic activity, which will decrease the activity of the stomach and the production of the stomach acids and stomach juices. If the stomach itself gets sort of to acidity, or if you have some sort of emotional stresses that activates the sympathetic nervous system, that can inhibit the activity of the stomach, either through those sort of autonomic activities or through the sort of lower production of that stimulatory gastrin hormone. And the intestinal phase is mostly a inhibitory phase. So I mentioned we get like a little bit of gastrin from the small intestine that gives us just one last little burst of secretory activity by the stomach, but very quickly that moves into activating sort of more inhibitory events. So as the small intestine gets distended, as it gets stretched, as it senses that there's that hypertonic, that sort of hyperacidic chyme, that there's foodstuffs in the small intestine, very quickly you're going to activate the enterogastric reflex, both long and short. So more of a neural mechanism that is going to inhibit the activity of the stomach. And you will also get the release of our enterogastrone hormones that inhibit the stomach activity. So this is figure 23.19. It's definitely a very good figure to review. From the stomach, we then move into the small intestine. 3 parts to our small intestine, of course. The shortest section is the duodenum, which is the first section. Then we have the middle section, the jejunum, and then the last section, the ileum. This is sort of your primary digestive and absorptive organ. Most of the chemical digestion takes place here in the small intestine, as does most of the absorption of the nutrients from our food. There are some propulsive events. Of course, we're talking about peristalsis to move the food through the GI tract and through the small intestine, and then also segmentation, so that the chyme sort of mixes with all of the enzymes that are here so that we can fully break down our food and absorb as much of the nutrients from our food as we possibly can. Because this is our primary digestion and absorptive organ, the small intestine has a couple of really important modifications. For one, the mucosa and the submucosa of the small intestine is folded, and there are these little circular folds. They're called plicae circulaires, and they essentially sort of force the chyme to sort of slowly spiral through the small intestine, as well as helping to increase our surface area. We also then have the finger-like villi, which increase our surface area even more so that we can pack lots of absorptive cells into the lining of the villus. And then on the very top of that villus, you will find what we call microvilli or brush border, which is going to contain enzymes that will help us break down our food fully. So these three modifications, the plicae circularis, the villi, and the microvilli, increase the size of the surface area of your small intestine about 600 times its other size. It's about the size of a tennis court. So you basically have a tennis court in terms of surface area in your small intestine, which is a lot. The chyme from the stomach, right? We've got partially digested carbs, partially digested proteins, mostly undigested fats, a very low pH from the stomach acid, and hypertonic, so lots of stuff in it. Most of the enzymes from the that are going to kind of do stuff here in the small intestine are going to come from the liver and the pancreas, but then we also have sort of some finishing enzymes from the brush border. Typically it takes about 3 to 6 hours for your small intestine to absorb, to digest and absorb all of your nutrients from your meal, as well as most of the water from your meal. So it's not a super fast process, it's not a super slow process, but we want sort of maximal time so that we can really kind of wring every last bit of nutrient from our food. Chyme delivery from the stomach into the small intestine is quite slow. You would not want sort of all of the food from your stomach to immediately go into the small intestine because it's hypertonic. It would essentially suck all the water from your blood. You also need to adjust the pH upward because it's too acidic. You want lots of time for the chyme to mix with the bile and the juices from the pancreas, and you really just don't want to get that first part of the small intestine from sort of being overwhelmed. And so there are some reflexes in terms of the neural system, as well as some hormones that help us sort of regulate this activity. So initially, so right after you have eaten a meal, primarily what your small intestine is going to be doing is segmentation. So as it receives a little bit of chyme from the stomach, it's going to mix that with the bile from the liver and the pancreatic juices, and sort of just mix it all together. Allow the enzymes to break down the foods, and then between meals it's more peristalsis, so that we're sort of moving that mass of pancreatic juices and bile and chyme and food and partially digested stuffs through the small intestine. The small intestine ends at the ileocecal sphincter. That's what connects and separates the ileum from the large intestine. Several divisions of the large intestine. The first is the cecum, then into the ascending colon, transverse colon, descending colon, ultimately into the sigmoid colon, and then the rectum and anal canal. The ileocecal sphincter generally remains closed, but it will open whenever it is stimulated to do so by your sort of enteric reflexes or some hormonal control. There are lots of bacteria in your large intestine. Literally, you have like over 1,000 different species of bacteria in your large intestine. You actually have more bacterial cells in your large intestine than you have your own cells. Typically, they're going to come from the small intestine or from the anus. But bacteria are actually good for us, at least as long as they're not pathogenic. They do some fermentation for us, so some of the bacterial flora digest some of the things that we can't, that we don't have enzymes to digest. There's also a little bit of vitamin B and vitamin K synthesis that the bacteria do for us. And essentially by sort of taking up space in our large intestine, they sort of keep the pathogenic bacteria in check. So there's sort of no room for the pathogenic bacteria, since all of the spaces are sort of taken up by quote unquote good bacteria. There is sort of mounting evidence that proper sort of balance of your bacterial flora can affect your mood, can affect your weight, can affect even your susceptibility to different diseases like diabetes or arteriosclerosis. So there's certainly an avenue to explore in terms of sort of managing our healthcare in the future. In terms of the processes that go on inside the large intestine, there really isn't any digestion that occurs here except what the gut bacteria do. By this point, sort of most of your food itself has been digested and absorbed by the small intestine. There is some absorption of vitamins that take place in the large intestine, electrolytes that take place in the large intestine, and water. So this is sort of our last chance to wring whatever little bit of water out of our juices and our food and our drinks that we can. And so the large intestine sort of acts like a big old drying oven. Typically the residue remains in your large intestine for about 12 to 24 hours. It uses propulsion and peristalsis to push that residue through the large intestine, ultimately to the anus in order to be defecated. The last segment of the large intestine is the rectum. It opens to the exterior of the body at the anus. There are two internal sphincters and an external sphincter, two sphincters, an internal sphincter and an external sphincter. The internal sphincter is involuntary, smooth muscle. You don't have control over that one. The external sphincter is skeletal muscle. You do have control over that one. So this is what defecation looks like. Essentially, as the feces sort of moves into the rectum, sort of starts to stretch out the rectum, that activates some stretch receptors there. That will activate the sort of defecation reflex, a nice little spinal reflex. And that will lead to the relaxation of the anal sphincter. However, of course, you're getting sort of impulses from the cerebral cortex as well to say, oh, hey, intestine tells me it's time to get rid of these feces, but it's not really a good time to do that. And so you will maintain control of that external sphincter until it is a proper time to defecate, and then you can pass your feces. If you've ever done what they call Valsalva's maneuver, it's when you sort of close your glottis, you sort of contract your diaphragm and your abdominal wall, sort of puts a little bit more pressure there on the defecation process. That can help you to pass your feces more easily.